Hi there, it's Mrs. Vance again. This is Unit 7 Notes, Part 4, and today's topic is the process of mitosis, which is how eukaryotic cells carry out cell division. So cell division it involves a continuum of changes. In other words, it's throughout the whole life of the cell. But the mitosis part itself is composed of 4 main stages. 5 if you count the one that's kind of in between. The 4 main stages are prophase, metaphase, anaphase, and telophase. Your book includes prometaphase, which is kind of in between prophase and metaphase. What you need to realize about the process is that it's basically a continuing straight on from prophase into metaphase. It's not like this stops and that starts. It's like a continuum. So most times you will not be responsible for the term prometaphase. We just use it in the notes because it is in your book in case you're reading something in the book. But if you're asked questions about it, you can be asked questions about prophase and metaphase and anaphase and telophase and not the other one. And then cytokinesis, which is the separation of the cytoplasm, often happens at the same time or right at the end of telophase. One thing that cells need when you have multiple chromosomes, we talked about in the last notes how simple it was for bacteria because they've only got 1 chromosome, but eukaryotes have multiple chromosomes. And some cells have only a few, like fruit flies have 8 chromosomes, not that many. We have 46, which is a pretty good number, but there are lots of other organisms that have even more chromosomes than we do. And so it's important to have a way to organize the chromosomes to make sure that they're equitably distributed to the daughter cells. Each daughter cell needs to have 1 copy of each chromosome, and so you have to have a way to do that. And in cells, this is a structure we call the spindle. It is composed of microtubules, and is produced by the microtubule organizing center of the cell called the centrosome. That basically organizes the cytoskeleton of the cell that involves microtubules. In animal cells, we have a pair of structures called centrioles that are present in the centrosome that help organize and distribute the microtubules properly. And so we'll refer mostly to animal cells when we talk about the spindle because we'll show how the centrioles are involved in that. So spindle is important. So here's what happens in each of the phases. In interphase, again, the cell is doing its normal job, and it's also getting ready for cell division. So during the whole period of interphase, the cytoplasmic contents are going to at least double in size, and 2 centrosomes are going to form, containing those centrioles in animal cells. The chromosomes are going to duplicate in the nucleus during the S phase. Once it's received the signal, it's time to get ready for division to occur. But you still can't see, when you look at this picture of the cell, you can't really see the individual chromosomes, but replication has occurred. And the nuclei, as we're getting ready for cell division to begin, the nucleolus, which is visible now, is going to start disappearing in just a little bit. So in interphase, you can see the nucleolus. Many cells have more than 1 nucleolus, and so you can see multiple ones sometimes, depending on the view of the cell. And everything is getting ready to go. That's an interphase. Once the signal starts for the mitotic phase, we'll start to see some changes. So right now here we see a photograph of the same cell. Again, you can look at the nucleus there. You can't really see anything in particular. The yellow strings here are the microtubules. Here are the centrioles, the centrosomes that are organizing those microtubules. And we can't really see anything in particular going on there. It's just what the cell looks like under this particular process. In prophase, the microtubules begin to emerge from the centrosomes and start forming the spindle. In the nucleus, the chromosomes coil up more tightly and become compact, and the nucleolus or nucleoli disappear. So that's what the first changes that we start to see in prophase. In prometaphase, as we continue on toward metaphase, the microtubules are going to reach the chromosomes, and they're going to attach the centromeres of the sister chromatids and start moving the chromosomes to the center of the cell. The nuclear envelope disappears, and other microtubules meet the ones from the opposite poles, and so they form a whole structure. So here's our our prophase and prometaphase as we go from one to the next, okay? So here's our early spindle starting to form. You can start to see the chromosomal bodies. The picture over here, you can see there's definitely two centrosomes that are moving apart from each other. You can definitely see the yarn-looking structures in there, the wiggly things that look like little worms or something like that in the nucleus. Those are the chromosomes, but it's still contained within the nuclear envelope here. And as we progress toward prometaphase, the nuclear envelope starts to break apart or be reabsorbed. The chromosomes continue to condense and get smaller, and the microtubules from the spindle attach to the kinetochores, which are part of the centriole, that are centromeres that attach the sister chromatids to each other. And so all of this stuff is happening here in the picture. You can see definitely you've got your two different centrioles there, the spindle formed in between them, with the microtubules attaching to the chromosomes, and the chromosomes moving out of the nuclear area and gradually moving toward the center of the cell. Metaphase, meta means middle, okay? And so in metaphase, you've got the fully formed mitotic spindle. You've got the chromosomes that line up at the cell equator, because basically they've moved to the middle of the cell. And then the kinetochores of the sister chromatids are facing opposite poles. The kinetochores are places where the microtubules attach, and they're facing the opposite poles of the spindle. So let's look at some pictures here. Here's our chromosomes, the imaginary equator there, the metaphase plate, okay? And we see our spindle fully formed there and attached to the chromosomes. In the picture, you can see now, you see the fully formed spindle, and all of the chromosomes are attached to the spindle fibers there, and everything is set and ready to go. Once everything is ready to go, then we're ready to move into the next phase. Okay? In the next phase, anaphase, this is when the sister chromatids separate at the centromeres. There are enzymes that will actually cut apart right there, that are the strands that are holding the two chromosomes together. And the daughter chromosomes now are moved to opposite poles of the cell. The microtubules of the spindle kind of act like they're doing a tug of war, and so when the centromere separates the two chromosomes, it's kind of like someone cuts the rope, and both sides of the tug of war pull to their opposite ends. At the same time, you've got the cell elongating, because they've got other microtubules that are extending from the centromeres that are actually pushing against the ends of the cell, pushing them longer and elongating the cell. And so the chromosomes get moved from the center of the cell toward the poles. So here we have the centromeres the centrioles have separated, and the chromosomes are being pulled to opposite poles, and the cell is getting longer as that is happening, because these microtubules up here at the ends are pushing against the ends of the cell, elongating it as the chromosomes are separated from each other. The last phase is telophase. In telophase, the cell continues to get longer. A nuclear envelope starts to form around the chromosomes. The chromatin starts to uncoil. The nucleolus reappears in each of the chromosomes, in each of the nuclei, and the spindle starts to disappear. And you can see that happening here in the diagram. You can see right now, you can see that cytokinesis is starting here, as it usually does during telophase. The chromosomes are unloosening up, basically. The nuclear envelope is starting to reform around them. They're condensing there in the middle of the cell. And then you start to have this cytokinesis happening that's going to separate the cells into two separate daughter cells. You can see the same thing happening over here in the photograph. During cytokinesis, that's when the cytoplasm gets separated, and there's a couple of different ways it happens depending on whether you're talking about plant cells or animal cells. There are other things other than plants and animals, yes, but those are the main things we talk about when we talk about cell division. In animal cells, cytokinesis happens as a result of a cleavage furrow. Cleavage furrow is kind of like a drawstring. There's motor proteins that actually can tighten there. And so think about when you pull the drawstring in your shorts, how that tightens the waist of the shorts. If you pull the string there, that's the microfilaments, the contracting ring around the cell, that's going to start separating the cells into two parts. The cleavage furrow is going to deepen in order to separate the contents of the cells. So basically you have like a drawstring that's being pulled here to tighten, and if you look at the diagram, you can see it's a contracting ring of filaments that are pulling together and eventually going to separate the cell into two separate daughter cells that are more or less equal in size. So there's the cleavage furrow. You can't really see the contracting ring of microtubules because that's on the inside, but basically that's the thing that's happening, is that it's separating the cell, the big cell, into two smaller cells. In plant cells, since there is a cell wall, you have to have a slightly different mechanism. And so a cell plate forms in the middle of the two nuclei from vesicles that are produced that contain cell wall material. The vesicles are membrane-bound structures containing the cell wall material, mostly made of cellulose. And the cell plate grows outward to reach the edges. So basically what happens is all those vesicles collect there in the middle along the metaphase plate and start to coalesce with each other, forming a cell wall in between two new membranes. And so now we have each cell with a plasma membrane and a cell wall, like you see here. Here's the cell plate forming. And you can see in the diagram, you can see the cell plate forming there. Those are vesicles containing the cell wall material. And as they grow and coalesce together, that's going to make the cell plate, which is going to continue to grow outward toward the wall, toward the cell membrane and the cell wall. And now the membrane that contains the vesicles is going to fuse with the membrane of the cell to form totally separate cells from each other, containing a cell wall in between them, in between the cell membranes. So just for a little closer view here, we have our daughter nuclei forming there. There's the cell plate starting to form and growing from the center out toward the edges. Again, in the diagram to show you how those are coalescing together to make our two brand new daughter cells that are now smaller than that original cell was, but that can continue to grow and get larger like the parent cell did. Just a quick review. Let's go back over the diagrams once again. So in interphase, you can't really see the chromosomes. There are microtubules present in the cell, but they're not organized in the same way as they will be for cell division. Just before cell division starts in the S phase and the G2 phase of the cell cycle, the chromosomes have been copied. Everything is ready to go for cell division. When you enter the mitotic phase, that's when the chromosomes start to coalesce or condense down into their more chromosome-like structure from the loose chromatin formation. You've got the spindle starting to form. You've got the nuclear envelope starting to break down. And as you move from prophase into prometaphase, you see the nuclear envelope is disappearing. The chromosomes have tightened up even more and are attaching to spindle fibers and moving to the center of the cell. Here we have in metaphase, we have the chromosomes on the metaphase plate, ready to be separated. And as you go into anaphase, the centromeres split and the daughter chromatids or sister chromatids are pulled apart. The cell elongates. And then in telophase and cytokinesis, you have the cleavage furrow forming, the new nuclei forming, the DNA loosening back up into its chromatin form and ready to go to form two brand new daughter cells.